Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So, we were discussing about immunology, where we first learned about the immune system, the what is immunity, and what are the different types of immune systems that our body possesses. So, we learned about the innate immunity that we possess since the birth, and we also learned about adaptive immunity that develops over the time due to the exposure to different pathogens due to the different experiences that we have during our lifetime, right? So in the innate immunity, there is the cellular part of the innate immunity as well as the humoral part of the innate immunity, right? There are innate immune system cells as well as the humoral component is the complement systems, the cytokines, interferons, right? So in the same way, adaptive immune system also has its cellular component as well as its humoral component. The cellular part contains the B cells, T cells, those lymphocytes, and the humoral part contains the antibody secretion by the B cells, right? So we just had a kind of introduction about innate immune system and adaptive immune system, but we'll be discussing about details about them in the future lectures, right? So then we discussed about hematopoiesis, the process of formation of blood cells from the hematopoietic stem cells, right? So we had a whole overview about how hematopoietic stem cells give rise to different progenitors that are going to give rise to the mature blood cells. So the hematopoietic stem cells, they are very rare and they are found in the bone marrow. So only one in the each 50,000 bone marrow cells we can find one hematopoietic stem cells, right? So this hematopoietic stem cell it has enormous proliferative capacity and it gives rise to the myeloid lineage as well as the lymphoid lineage, right? So we learned about all those stuff in detail. So when we started discussing about that, let me just have a quick recap about hematopoiesis. So hematopoietic stem cells give rise to the myeloid lineage as well as the lymphoid lineage. Right, the myeloid progenitor, the common myeloid progenitor cell that gives rise to the erythrocytes, erythroid progenitor, as well as the myeloid progenitors, right? So the myeloid progenitors, one of them is the granulocyte monocyte progenitor. So this granulocyte monocyte progenitor, this gives rise to the granulocyte neutrophils, and this also gives rise to the monocytes. Monocytes which further developed into macrophages, or they can also develop into dendritic cells, okay? Depending upon the conditions, they can either develop into macrophages or they can develop into dendritic cells, right? So this is the part that we need to focus. We first learned about the neutrophils, okay, which are the granulocytes. Then there are eosinophil progenitors, there are basophil progenitors, which are going to give rise to eosinophil and basophil respectively. So we learned about neutrophils, eosinophils, and basophils, as well as the mast cells. All of them, they are the granulocytes, means the leukocytes, the WBCs, which have granules in their cytoplasm, right? So today we are going to learn about macrophage in detail, which arises from the monocyte. Okay, so the macrophages are the phagocytic cells just like neutrophils, and they also has the antigen presentation job, so they also activate T cells just like dendritic cells. So it is quite clear from this chart, from this particular schematic, that the macrophage will resemble, because in the lineages it is closer to the neutrophils as well as the dendritic cells, so it is going to have some similarities with the neutrophils as well as the dendritic cells, okay? So just like neutrophils, macrophages, they are also phagocytic cells, and just like dendritic cells, macrophages, they are going to activate the T cells. So in the last class we learned about dendritic cells in detail, where we learned about the antigen presenting cells. So we discussed that besides the dendritic cells, which is the master antigen antigen presenting cell, because it is the one which has, which whose primary function is antigen presentation. So just like the dendritic cells, macrophage as well as the B cell from the lymphoid origin, both of them, they does this job of antigen presentation to the T cells, and this activates the T cells. So this initiates the adaptive immune response. So we can say that dendritic cells and macrophages, they are a link between the innate immunity and the adaptive immunity, because the innate immunity, the cellular part of innate immunity, is comprised of the myeloid cells, the cells of myeloid lineages, as well as the natural killer cells, which actually belong to the lymphoid lineage. So macrophages and dendritic cells, both of them, they are a part of innate immunity. So that they are kind of linking the innate immunity with the adaptive immunity, because they are going to activate T cells in future with the help of antigen presentation, right? But the primary function, antigen presentation is the primary function for the dendritic cells. For macrophages, the primary function is phagocytosis. So just like neutrophils, they are also phagocytic cells, but the difference is that neutrophils are very aggressive because they use, oxygen dependent killing, while the macrophages, they are not that aggressive, right? So we will learn about differences between neutrophils and macrophages as well. But before that, let's start learning about what is a macrophage, right? So this particular cell here, it is representing a macrophage. So this is a macrophage. You can see a lot of projections which are called as pseudopodia, because they are going to help in the phagocytosis. So those pseudopodia, here you can see those pseudopodia, they are going to engulf the particular pathogen or the bacteria which is present in the extracellular environment, and then that is going to form the phagosome. Okay, so this phagosome, when it merges with the lysosome, it is called as phagolysosome. So first the phagosome is formed, and then it forms phagolysosome. And in this phagolysosome, the microbial contents that they are going to get digested, the peptides, they are going to get digested. And once they are digested here in the phagolysosome, those digested peptides will, they are now antigens, and they will be presented over the surface of the macrophage, and then with the help of the MHC molecules, which macrophage possess, the MHC1 as well as MHC2 molecules, right? If you don't know about them, I also discussed them in last class, but I'll have a separate lecture about major histocompatibility complex as well. So don't worry about that. For now, it's what you need to know is that MHC molecules are the ones who are going to help in the antigen presentation to the antigen presenting cells, right? By the antigen presenting cells. So phagolysosome, in this phagolysosome, those antigens will be prepared, because the macrophage is engulfing the whole bacteria. So the bacterial components will be broken down, and now they are converted into short peptides. So those peptides serves as antigen for activation of immune responses. Okay, so those antigens that are now taken in, that are there in the phagolysosome, they will be, presented over the surface of the macrophage, like little flags for the activation of immune system. So that presentation, the antigen presentation over the surface of macrophage, that will be done with the help of MHC molecules. Okay, major histocompatibility complex. So we discussed about those in last class. So I hope you are getting the stuff. Now let's discuss about macrophage. How macrophages, they arise from the monocytes. Okay, so monocytes, they are a part of the white blood cells. So 5 to 10% of the white blood cells are monocytes. So they are rare compared to the, neutrophils. Neutrophils make up almost 50 to 70% of the WBCs, while the monocytes, they are only 5 to 10%. Okay, so those monocytes, they are found in the circulation in the blood. So let me just discuss what happens. So this is your bone. Okay, and in this bone exists the bone marrow. Okay, so this yellowish part is the bone marrow. So in the bone marrow, there exists the hematopoietic stem cells, which are going to give rise to the progenitor cells, right? So here will exist the granulocyte monocyte progenitor. So this yellow ones are the granulocyte monocyte Progenitors. Okay, so hematopoietic stem cells differentiates into progenitor cells in the bone marrow itself. Okay, so once it get differentiated into progenitor cells, these progenitor cells will get matured in the circulation to form the blood cells. So here now when they get matured, they will be circulated in blood. So in the blood they will form monocytes. Okay, so now the blood has monocytes, which are the matured cells, okay, which are mature cells, so which gets differentiated from the granulocyte, monocyte, progenitor. So those monocytes, they are in the circulation and they make up 5 to 10% of all white blood cells. So those monocytes, when they are in the blood and when they migrate to the peripheral tissue. Okay, so now upon migration to peripheral tissue, they have two choices. Either those monocytes, they can get differentiated to form macrophages, or they can get differentiated to form dendritic cells. Okay, so the maturation of those monocytes or the formation of macrophages or the dendritic cells, that will be seen in the peripheral tissue. Okay, so as I discussed in the last class, dendritic cells, they are the ubiquitous cells, means they are found in all over the body. So the tissue localization of dendritic cells is very strategic because they are the patrolling cars. So we need to put them at the hotspot locations where there's most where the need for dendritic cells is the highest. So they are found in blood, in skin, in spleen, thymus, right? So they are found in multiple tissues. In the same way, macrophages they are also tissue resident cells. So they are also found in tissues and they are found in multiple tissues. So most of times they are tissue specifics. So those tissue specific macrophages have their own names. So based on which tissue that macrophage is localized, we have different names for them. So the macrophages, if they are localized in the central nervous system, in the brain, in our central nervous system, then they are referred as microglial cells that we have discussed in detail in the neurobiology lectures, right? If those macrophages, they are there in the lungs, then they are referred as alveolar. Okay, they are referred as alveolar. Oh, oh. Okay, they are referred as alveolar macrophages. Okay, if those macrophages, they are there in the intestine, then they are referred as intestinal. They are referred as intestinal macrophages. Okay, if they are there in the bone, then they in the bone, for the bone homeostasis, means the formation and breakdown of the bone, the cells, the macrophage cells, they help them a lot, and there they are referred as osteoclast. So the osteoclast, if you might have learned this in your 12th standard, in your high school days, that osteoclast and osteoblast, both of them, they work in coordination for the bone homeostasis. So the bone breakdown cells, the cells which are going to do the phagocytosis, are the osteoclasts. Okay, so osteoclasts are the macrophages that are found in the bones. If the macrophages, they are found in the liver, then they are called as Cooper cells. Okay. The lung, the lung alveolar macrophages, they are also referred as dust cells because they're not just engulfing pathogens, but they also take care of the allergens such as dust. Okay. If the macrophages, they are found in any type of connective tissue, then they are referred as histiocytes. Okay, so these are the different types of different macrophages that are present in the tissues. So all of the type of tissues, almost all of them, they have macrophages because we need to have the cops everywhere to to maintain the to control the pathogenic attack, to protect our body from the pathogenic attack. Okay, so these are the types of the macrophages. So the macrophages, they get activated by pathogens or by cytokines. So the cytokines which are released by other immune cells such as dendritic cells or such as mast cells, they are going to alert those macrophages, or they can be activated by directly interacting with the pathogenic antigens. So let's assume that this is the pathogen, okay, which is arriving. So this is the site of infection, and it is been coated with some ligand, okay, the ligand for which the receptor is present over the macrophage. So these all of them, they are the molecules, the surface markers for the pathogen, okay, so they are present over the pathogen, okay. So this is how the pathogen is going to arrive at the site. And once it arrives at the site, the macrophage, which has its receptor, okay, for this ligand, which are distributed over the surface, though those particles will be received by those ligands will be received by the receptors. So this ligand receptor interaction, so this is the macrophage and this is the pathogen. So pathogenic molecules, okay, they are serving as ligand for the receptors present on the macrophage, right? So this ligand and receptor interaction, that is going to result into receptor clustering. Okay, this will result into receptor clustering. So those all of those receptors, they will be clustered in an area, and this receptor clustering will activate those receptors. So you remember when we were discussing about cell signaling, we discussed that how the dimerization or tetramerization of the receptor molecules is going to activate their intracellular domain, and it will activate the intracellular signaling cascade. In the same way, the receptor clustering is going to activate the macrophages. Okay, so this receptor clustering will activate the macrophages, and that will result into polymerization and depolymerization of the cytoskeleton. Right? We have discussed this in the cytoskeletal lectures. So this will result into polymerization and depolymerization of cytoskeletal elements that will form, a phagosome, okay, a vesicle where this pathogen will be taken in, and this phagosome will now merge with lysosome, which have hydrolases, the digestive enzymes, and this will form a phagolysosome. So in this phagolysosome, the degradation of this pathogen will be done. So once this pathogen is degraded, those molecules will be broken down into peptides, and those peptides will serve as an antigen for the activation of immune system. So those will be transferred to the MHC molecules, and MHC molecules are the ones which are going to hold those antigens over the surface of the macrophages. And this is how the macrophages, they are the phagocytic cells, right? First they do the phagocytosis, and they also does the antigen presentation. But the activation by antigen presentation, the activation of T cells by the antigen presentation by the macrophages, that is poor compared to the dendritic cells. And that is why if the question is that which is the antigen presenting cell, the main or the primary or the master antigen presenting cell, then we always choose for the dendritic cells, because they are the ones which are more, towards this job, who are prioritizing antigen presenting job over the phagocytosis job. The main way of killing pathogen by the macrophages is the phagocytosis. They also helps in activation of T cells, but the main job of them is the phagocytosing the antigen and killing them. So once they engulf this pathogen, rather than serving it as a peptide, as an antigen to the T cell, they will just digest it and degrade the whole pathogen, and they will end up killing the pathogen by themselves in some cases. So that is why we call the macrophages as the phagocytic cells. Okay, so those macrophages, they resemble to the neutrophils, because both neutrophils and macrophages, primary job of both of them is to do the phagocytosis. But besides those resemblances, the macrophages, they have differences. They differ from the neutrophils in some aspects. So we will now discuss about the differences between macrophage and the neutrophils. Okay. So as I told you, both of them, they are phagocytic in nature, but the viability of both of the cells is different. Macrophages, they are long lived. Okay, they can live for few days to months over to years because the tissue resident macrophages, they also have a long life. On the contrary, the neutrophils, they are short lived. Okay, they do not have that long life. Macrophages, they are tissue resident. Already discussed this. Okay, they are found in the tissues. While the neutrophils, they are not resident. They are found in the blood circulation. Okay, so they are circulating in the blood, and once the need is there, they are going to arrive at the site of infection first from the blood. So, if the question is that which are the cells which arrive at the site of infection from the blood circulation, then answer will be neutrophils. They are the first ones to arrive at the site of infection. So they are found in blood circulation. They are not found in the tissues. They are not localized in the tissues. The other difference between macrophages and neutrophils is that the macrophage, they can migrate from the tissue to lymph node for the antigen presentation. Okay, so they can migrate from tissue to lymph node for antigen presentation, but this is not seen in the neutrophils. Neutrophils, they do the diapedesis, means the movement from the blood vessels without rupturing those blood vessels to the interstitial tissue. So they are going to go there and they are going to do their job. They are going to phagocytose the pathogen, but they do not migrate from this tissue to the lymph node for the antigen presentation or for the activation of T cells. Okay, so this is not the job of the neutrophils. This is only seen by macrophages. Okay, so the macrophages, they get activated by cytokines. Okay, but they do not stimulate inflammation. So they do not, they do not activate the inflammation. So they are not strong stimulant for activation, for inflammation. Okay, whereas the neutrophils, they induce inflammation. Okay, so they are going to release some inflammatory mediators. So they does induce the inflammation. The macrophages, they are the phagocytic cells, but as I already told you, they are not that aggressive because there is no oxygen dependent killing. Okay, whereas the neutrophils, they use the oxygen dependent killing. They use the ROS production for killing the pathogen, and that is why they are very aggressive. This phagocytic capacity of macrophage is very high, but they do not use the oxygen dependent killing like the neutrophils. Okay, so they do not use they do not use the ROS production for killing the pathogens. So these are the differences between macrophages and neutrophils. Okay, so macrophage, they also resemble to the dendritic cells in the aspect of the antigen presentation, right? So when we discussed about dendritic cells, I told you that there's a factor which is known as GM-CSF, granulose. Okay, so this is a colony stimulating factor, and this granulocyte colony stimulating factor, this will be helpful for in vitro generation of the dendritic cells. So in the same way, there's one more factor known as macrophage colony stimulating factor. Okay, so this is macrophage colony stimulating factor. So this is going to help for the in vitro generation of the macrophages. Okay, so this is used for in vitro generation of macrophages. Okay, so so in vitro generation will be done with the help of bone marrow. So let me just discuss how do we do that. So what happens is that for the in vitro generation of macrophages, okay, so these are the these are the signaling chemicals. Okay, so these are the cytokines. So these are going to signal the cells to divide and to get differentiated into macrophage, right? So that for that we need the bone marrow cells. So in the bone marrow cells exists cells, the hematopoietic stem cells, which are going to get differentiated into any of those cells, macrophage, the granulocyte, or the dendritic cells, right? So that is why we need bone marrow for in vitro generation of macrophages. So we take out bone marrow from mice. So we need to follow a certain procedure, a certain repertoire for that. So first we take the mouse, okay, and we sacrifice the mouse according the ethical guidelines, okay, using the appropriate ethical guidelines. We sacrifice the mouse. Okay, once we have sacrificed the mouse, we take out the longest bone in the mouse, which is femur, from the hind leg. So we take the femur from the hind leg and we dissect it. So we dissect the femur, okay, which is taken from hind leg. We take the longest bone because bone marrow will be present there, right? So once we dissect this femur, once we cut off the ends of the femur, so this is femur. Okay, so once we cut off the ends of the femur, what we do, we use a syringe with a needle to flush out the bone marrow. Okay, so then we flush out the bone marrow using a syringe with a needle. Okay, using a syringe with a needle, we flush out this bone marrow. So once we have flushed out this bone marrow, we are going to disaggregate the cells. Okay, we are going to break up the clumps which are present there. So we will disaggregate the cells. Okay, we will gently break all of the clumps and we will ensure that we have got the single cell suspension. So we can also use the cell strainer for straining the cells, and we can have, so this all is to ensure that we have a single cell suspension. And once we have the single cell suspension, we are going to centrifuge it and resuspend it, and then we add the those factors, okay, according to the need. So when we add the macrophage colony stimulating factor, MCSF, and we keep it, we incubate this cell suspension for a few days, okay, so for, let's say, for 10 days, then we are going to get the macrophages. Okay, so this is how, with the use of bone marrow and with the use of those factors, we are going to generate them in the in vitro conditions. Okay. So whereas the granulocyte macrophage stimulating factor, colony stimulating factor, this is the one which will be helpful for the, for the in vitro generation of dendritic cells. And this is also used for enhancing the engulfing capacities of the granulocytes or of the macrophages. So this can be used as the factor to increase the efficiency of those phagocytic cells, whereas the macrophage colony stimulating factor is necessary for the in vitro generation of the macrophages. Okay, so this is the process. This is how we use the bone marrow from the mouse cells to generate the macrophages. Okay, so with this, I'm done with the lecture. I have discussed about macrophages. Okay, there's one more thing called opsonization that will be beneficial to discuss here. So let's discuss what is opsonization. Okay. Hmm. So what happens is that those macrophages, they, as I discussed previously, these macrophages will have receptors for the molecules that are present over the pathogens. Okay, but besides those receptors for the ligands present over the pathogens, macrophages also have receptors for the antibodies. Okay, so they have the antibody receptors. So let's say that the antibody is IgE. So they are going to have anti-IgE receptors present over their surface. Let's assume that the antibody is IgG. So they are going to have anti-IgG antibody receptors over the surface. So this is just an example. Okay, so it means that the macrophages, okay, they have antibody receptors, means the receptors which can receive the antibodies, which can bind to the antibodies. Okay, so besides the macrophages, as discussed earlier, they also have the receptors for the ligands present over the pathogens. Okay, but they also have the antibody receptors. So when those ligands bind to the receptors over the macrophages, let's call those receptors as the LR, the ligand receptors, and let's call those antibody receptors as AR. So the killing of pathogen, which is mediated by the receptor clustering, the LR clustering, that is weak compared to the antibody receptor clustering. So this antibody mediated killing of the pathogen, this is 400 times more effective than this receptors for the ligand clustering. Okay, so what happens is that the pathogen molecule, okay, this is the pathogen which has multiple uh, ligands over it. Okay, let me draw it properly so that there will be no confusion. So this is the pathogen. So this has multiple. Okay, so this has multiple ligands over it. Okay, so those ligands can bind to their particular receptors over the macrophage as well. But the antibodies which are present there in the circulation, in the tissues which are being released there, okay, so this antibody will also have the capacity to bind to those ligands. So when this antibody bind to those, okay, so I'm just drawing it simply as Y shaped. Okay, so these are the antibodies bound to those receptors. So this antibody coated pathogen, so when this antibody-coated pathogen is received by the macrophage, the efficiency of killing this pathogen will increase. Okay, so instead of this simple pathogen which has the ligands coated over it, and for those ligand receptors are present on the macrophage, this is the case one. Okay, that this will be engulfed by whole by the macrophage, or this is the case 2. So in the case 2, the killing efficiency is high, it is 400 times higher than the case 1, because those antibody coated pathogens, they will be received better by the macrophages because of the abundance of the antibody receptors over the surface. So this macrophage has a lot of antibody receptors over it. So those antibody receptors, okay, which are present there on the macrophage, they will be receiving they will be clustering, and that is going to activate the macrophage, and that will result into the pathogen killing. So this particular process, the modification of pathogen with a molecule which is referred as opsonin, is known as opsonization. Okay, so in this case, the antibody molecules, they are opsonin molecules. Okay, so for that, let's discuss what is opsonin. So opsonin molecules are, they are the ones which are going to help the pathogen recognition, which are going to, make the immune system better at visualizing those pathogens. Okay, so for understanding this, let's take an example. Let's assume that you are going to go and visit your relative in some other city, right? So you are leaving city A and you're flying over to the city B to meet your friends, to meet your relatives. Okay, and that is why they are going to, so they are going to send a driver to pick you up at the airport. So the driver doesn't know you, right? He has never seen you before, and you haven't seen the driver either. So when you are there at the airport, the driver will be waiting for you in the lobby, holding a board with your name, right? And that is going to make both of you to, that is going to make you able to identify, okay, this is the driver which is sent for me, and this is going to take me to my location. Okay, so this is how that little board that is helping you to recognize that driver is known as opsonate. In the same way, those antibodies, when when they bind to the pathogen, they are like a little boards which are present on that pathogen, and they are making immune system able to recognize this pathogen. So they are making, they are enabling the macrophages to identify this pathogen whenever antibody is there, because it makes it easier, right? Whenever there's board, okay, you don't need to, there will be no need to go and ask each person, okay, are you sent by that my friend, are you sent by my relative, right? So it just makes your job easier by having that board in the hand of that driver, right? So you saw your name and you are just going to, you will go straight to that person, you will say that, hi, I'm this person, and let's go to our location, right? So this makes it easier for you to identify that driver. In the same way, opsonin molecules are the molecules which are making the which are enabling our immune system to visualize those pathogens better. So these are the modifications which are added over the pathogens. Opsonin molecules are the molecules which are added over the pathogen molecules to make their recognition better to the immune system. And this whole process of modification of pathogen with the opsonin molecules is known as opsonization. Okay, so I hope opsonization is clear to you all. But besides the killing, besides making it recognizable, besides making the pathogen recognizable to the immune system, to our immune cells, besides this job, opsonization also has other jobs. So we will be discussing about those roles, the roles of opsonization besides this in detail in the subsequent lectures. Okay, so for now, I just introduced the idea of opsonization to you all. So we are done with the macrophages. We have discussed them in detail. In next lecture, probably we'll discuss about the natural killer cells. So for for summarization, we have discussed about the granulocytes, okay, the neutrophils, basophils, eosinophils, and we also discussed about mast cells, which are the tissue resident granulocytes. Then we discussed about the dendritic cells, which are the antigen presenting cells, which also belong to the myeloid lineage. And we now discussed about macrophages. So we are almost done with all of the myeloid lineage cells. So we are going to move to the lymphoid lineage, and we will first discuss about the natural killer cells. And once we have discussed about natural killer cells, then we'll discuss about the cells of adaptive immunity, the B cells and T cells, and then we'll proceed with the discussion about the immune organs, and then we'll discuss about the innate immunity and adaptive immunity respectively. Okay, so I'll catch you in the next lecture. Till then, see you. Bye bye.